Welcome to the Explainer. If you're revising for your GCSE biology paper, you're in exactly the right place. Today, we're going to tackle a really important core practical, the food tests. Now, this is required practical activity 4 on the AQA spec, so let's just break it down and make sure you know it inside out for your exam.
Okay, so here's our game plan. We're going to start with the basics of why we even do these tests. Then we'll dive into the method and the results for carbohydrates, proteins, and lipids, one by one. And to finish off, we'll wrap it all up with the key facts you absolutely have to remember to ace your exam.
Right, first things first. Before we get into all the test tubes and chemicals, let's get our heads around the why. What is the big question we're actually trying to answer with this practical?
I mean, think about it. A piece of bread or a slice of cheese. It looks pretty simple, right? But on a molecular level, it is packed with all sorts of different biological molecules. So how can we scientifically prove what's in there?
Well, the secret is using something called a qualitative reagent. Now, that word qualitative, that is a key term for your exam. It just means we're testing for the quality or the type of substance, like is sugar here, not the quantity, so not how much sugar is there. You can think of these reagents as little chemical detectives that change color to give us a simple yes or no answer.
So, for your AQA GCSE, you need to be able to test for these three major players: carbohydrates, proteins, and lipids. These are the molecules this whole practical is designed to find. Let's kick things off with the first one, carbohydrates.
Okay, we're starting with carbohydrates, but this is actually like a 2-for-1 deal. We need one specific test for the simple sugars and a totally different one for the big, complex carbohydrate starch.
So first up, sugars. The test you've got to know is called the Benedict's test. And, surprise, surprise, it uses a chemical called Benedict's reagent.
The method itself is pretty straightforward. You prepare your food sample, which usually means crushing it up and adding a bit of water to make a solution. Pop that into a test tube, add a few drops of the Benedict's reagent, and then, and this is the crucial step people forget, you have to heat it. A hot water bath is the safest way to do this in the lab, and you need that heat for the color change to actually happen.
And now for the results. This is the cool part. Benedict's reagent starts off as a bright blue color. If there's no sugar, it just stays blue, even after heating. But if sugar is there, you get this fantastic color change. It can go green if there's just a little bit of sugar, yellow for a medium amount, and a strong brick red for a high concentration. You really want to remember that brick red color. That's the classic sign of a strong, positive test.
Okay, so that's sugar's sorted. The other carbohydrate you need to be able to find is starch. And for that, we use a simple iodine solution. Honestly, this test couldn't be easier. You just take a small amount of your food, add a couple of drops of iodine right on top of it, and that's literally it. You're looking for an immediate color change. And a massive bonus for this test, no heating required. It's super quick.
So here's the change you are looking for. Iodine solution is a kind of browny orange color to begin with. If there's no starch, it just stays that color. But if starch is present, it instantly turns this deep, deep blue-black color. It's a really dramatic and obvious result. You can't miss it.
Right, we have absolutely nailed carbohydrates. So let's move on to our next key food group, proteins.
To test for proteins, you need to know the Biuret test. And this test, well, it uses a chemical called Biuret reagent. Simple as that.
The procedure here is just as straightforward as the starch test. You just add your food sample to a test tube, add the Biuret reagent, give it a little swirl to mix it all up, and then you just watch for a color change. Again, no heating needed for this one either.
So what are we looking for? Well, the Biuret reagent starts out blue. If there's no protein in your sample, it's just going to stay blue. But if protein is present, the solution turns from that blue to a really distinct purple or lilac color. That's your positive result right there.
All right, time for our final food group. We've done carbs, we've done proteins, now it's time to hunt for lipids, which, we usually just call fats and oils.
Now, the test for lipids is a little bit different. It's not really about a color change, it's about creating something called an emulsion. Just think about what happens when you try to mix oil and water for a salad dressing. They don't really mix, do they? You get that cloudy mixture. That's an emulsion, and that's the principle we're going to use here.
This method has a couple more steps, so listen up. First, since fats don't dissolve in water, we need to dissolve them in something else first. For that, we use ethanol. So you add your food sample to a test tube with some ethanol, and you give it a really good shake to dissolve any lipids. Then, and this is the key part, you pour that ethanol solution into a second test tube that's just got plain water in it.
And here is what you'll see. If there are no lipids present, the solution will just stay clear and boring. But if lipids were dissolved in that ethanol, the moment you add it to water, they form millions of tiny droplets, creating this cloudy, milky white layer. That is your emulsion, and that cloudiness is the positive test for lipids.
Okay, we have gone through all the methods and all the results for every test. Now let's bring it all together and really focus on what you need to remember to absolutely smash this in an exam.
This slide right here is your ultimate revision checklist. Let's just run through it one more time. For sugars, it's Benedict's, which goes from blue to brick red. For starch, it's iodine, going from brownie orange to blue-black. For protein, it's the Biuret test, which turns from blue to purple or lilac. And for lipids, it's the emulsion test, which goes from clear to a cloudy white. And the final, major point, remember, these are all qualitative tests. They only show if something is there, not how much of it.
And finally, just remember that in an exam, they're not only testing your memory. This practical is designed to test your actual skills. The AQA spec says that by doing these food tests, you're showing you can safely use equipment like water baths, that you know how to use these qualitative reagents properly, and that you can make and record accurate observations.
And that is everything you need to know for required practical 4 food tests. You are now fully equipped to be a food detective. The next step in this whole story, of course, is what happens after we eat these foods. How does your body actually use enzymes to break all these molecules down? But we'll save that for another explainer. Thanks for watching, and good luck with your revision.